So this is chapter 12, the senses. So this is a little less heavy than chapter 11 is overall. So initially we'll talk about the breakdown in general and special senses. So general senses are those such as temperature, pain, touch, stretch, and pressure. The receptors for general senses are mostly in our skin, our muscles, our tendons, our joints, and our organs. We have special senses as well, which includes taste, or the scientific term is gustation, smell or olfaction, our vision, our equilibrium or balance. Those are somewhat interchangeable, and hearing. So we have senses in order to detect and respond to stimuli, not just respond to stimuli, but respond appropriately to stimuli. And our senses also help us maintain homeostasis. Okay, so we have a few areas of the brain that are specifically dedicated to our special senses. So we have a primary gustatory complex. So remember, gustation equals taste. So that's the area of the brain dedicated to taste. We have a primary visual cortex. This is in our occipital lobe. If you remember, that occipital lobe in the posterior portion of the brain is our vision center. So that has our primary visual cortex as well as our visual association area. The temporal lobe contains our primary auditory complex. We also have an auditory association area in the temporal lobe. And then down there, kind of on the underside, you can see the olfactory association area. That interprets the sense of smell, and that is near the cranial nerve for our sense of smell. Okay, so our sensory receptors, like I said, you can find some in our muscle and tissue as well as our organs. So they form, as a group, can form a sense organ. It's specialized nerve cells or nerve endings, so they're specifically made to be able to sense whether it's taste, vision, whatever. They're very specialized cells. So the initial sensation or stimulus is detected by the receptor itself. If that stimulus is strong enough, that receptor is then going to transmit that information to the brain, spinal cord, our central nervous system, and that's when we actually get to have the sensation itself, such as sight or sound. So the brain actually filters out a majority of the stimuli. If you think about it, we have a lot of stuff going on all the time. If you can kind of imagine yourself in a busy city or on a city street, think if you were going to kind of try to tune in, or your brain was going to try to interpret every single sound you heard, you would drive yourself nuts. So your sensory receptors and your brain decide to focus on the important sensations, and a lot of it gets filtered out because we don't need to think about it. It's unnecessary. So our sensory receptors transmit the type, location, and the intensity of the stimulus. As I said, these are specialized receptors. So each sensory receptor is specialized for a specific type of stimulus. So our chemoreceptors are going to detect chemicals. This can include odor, taste. Of course, if a chemical actually is going to make contact with our skin, the chemoreceptors are going to have a role in that. Our mechanoreceptors, those are our pressure, stretch, stuff like vibration. Thermoreceptors is temperature. Nociceptors, the simple explanation is pain, but this is also related to damage from trauma. Heat can cause nociception. You can have pain due to chemical interactions. You can have pain due to lack of oxygen. So the big term is that nociceptors detect pain, but there can be multiple reasons for that. Photoreceptors are sensitive to light or detect light as a stimulus. These are only found in our eyes. And then proprioceptors are found typically in our muscles and tendons, as well as our joints, and they can detect muscle stretch, movement, orientation sensing. So what that means is our ability to tell where we are in space. All right, first review question here. Nociceptors respond to A, various chemicals inside and outside the body. B, pressure, stretch, or vibration. C, pain from tissue damage. Or D, changes in temperature. So C, pain from tissue damage. Remember, nociception is the detection of pain. Okay, diving a little further into our general senses. Remember, this is pain, pressure, touch, stretch, temperature. All of those are our general senses. And so these receptors are widely distributed. They can be the skin, the muscles, tendons, joints, our organs. Viscera is another name for organs, remember. So going back to pain. Remember, pain is detected by nociceptors. So these are abundant in the skin, our mucous membranes, and almost every organ. So you guys do not need to memorize or worry about the difference between fast and slow pain fibers. But our fast pain fibers are abundant in our skin and mucous membranes. That's our sharp stabbing type of pain. Like you stub your toe, you're super mad, that initial kind of really, really intense pain, that's your fast pain fibers. So our slow pain fibers, that's more of our deep body organ structure, produce a dull aching pain. For any of you that have experienced appendicitis, that's that kind of pain. Or even just like a stomach ache type of pain, that's your slow pain fibers at work there. Fun fact, the brain does not contain any nociceptors, so we cannot, or we do not actually detect pain in the brain. Okay, so our pain pathway here, I just put a link to our animation from the book there. All right, so the book breaks it down into 6 steps total. So initially the injury happens, and those tissues release chemicals that actually cause the nociceptors to react and send that impulse to the brain. So when our nociceptors create an impulse, that pain signal is going to go to the dorsal horn. Remember, that's where our sensory information goes. And from that dorsal horn, it'll then travel up the spinothalamic tract to the thalamus. Remember, I don't need you guys to remember the spinothalamic or the spinoreticular tract that we're going to talk about. But no, ascending tracts are sensory and descending tracts are motor. So we're talking about the pain pathway, that's a sensation. So these are both going to be ascending pathways. Oops, sorry, guys. Okay, so it goes from the dorsal horn. Remember, that's our sensory horn. That's going to go up the spinothalamic tract. That'll go from the spinothalamic tract, which will end in the thalamus. So that's one aspect of the pain pathway. While this is happening, our spinoreticular tract is also going to carry pain signals to the reticular formation. Remember, that's a part of the brainstem. So as we said, our thalamus got the signal from the spinothalamic tract. This will take it to the postcentral gyrus of the cerebrum, and that's when we actually become aware of pain. So the impulse from the spinoreticular tract goes past the thalamus, that goes to the hypothalamus and the limbic system. Remember, these are our areas of emotional response. And that's kind of the last aspect. That's what's going to cause our response to the pain. So in terms of the pain pathway and what you need to know, remember that pain will go to the dorsal horn because that's our sensory horn. You need to know that pain is a sensation, so it'll go up the ascending pathway. But in terms of memorizing the spinothalamic and spinoreticular aspect, you don't need to know that part. You should know that the thalamus, the hypothalamus, and the limbic system are involved in pain, but you don't need to know any other specifics outside of that. Okay, referred pain. This is more of a concept than anything. So remember we talked about the slow pain fibers being our deep type of pain. Um, so this is what this type of pain is going to travel over, is going to be our slow pain fibers. So basically what happens is when there's pain coming from an organ, you'll see all these different colors and areas have an organ associated with it. So when there's pain coming from an organ, oftentimes we'll actually experience it in a little bit different part of the body, and we'll feel it as more superficial. So this is known as referred pain. And so the reason that this happens is that kind of nerve pathways are shared and different areas of innervation are shared. So even though we're the pain source is in one area, the pain pathway and the pain pattern can be slightly different. So all you guys need to know about referred pain is that it can make it somewhat difficult to make a diagnosis. So a good example of this for myself as a physical therapist is, say someone is coming in and complaining of left-sided neck pain. If it doesn't really seem like any movement of the neck or any muscles of the neck are really bothersome, then I may have to consider the fact that their lung or diaphragm may be causing the source of their pain, even though they're feeling it up in the left side of their neck. So you should know that referred pain makes diagnosis difficult somewhat. And you should also know just the concept that referred pain means that you're feeling it in an area different than the pain source. Okay, this leads right into our second review question. So the true source of pain is tough to identify because A, nociceptors can be stimulated by a variety of chemicals. B, deep body organs do not contain pain fibers. C, some pain signals travel to the thalamus, whereas others travel to the limbic system. D, sensory impulses from different areas often travel to the brain over the same pathway. So the answer there is B, sensory impulses from different areas often travel to the brain over the same pathway. Remember, sometimes those nerve pathways kind of overlap. So some of the deep organs are supplied by the same areas that run superficial as well. So there is a chemical release when we have injured tissue, but that doesn't have to do with our source of pain. Those deep body organs have our slow pain fibers. And it is true that some pain signals travel to the thalamus while others travel to the limbic system. But that doesn't mean that we're going to feel it in a different area or that it's going to be tough to identify. Okay, we just talked about our general senses. Moving on to the special senses. This is taste, smell, hearing, equilibrium / balance, and vision. So again, we're talking about specialized receptors, and these are generally in specialized organs in clusters as well. Okay, so initially we're going to talk about taste, or gustation is the scientific term for it. So all over this diagram, you'll see the term papillae. So these are kind of protrusions on the tongue. And the reason that these protrusions are important is because a lot of our taste buds are located all around these protrusions or papillae. So there are 4 different types of papillae. I do not need you to be able to identify the exact function of each different type. What I want you to know about the papillae is that they are protrusions and that taste buds follow them a lot. This is a zoom in on our taste buds. So we have chemicals that are in our saliva that actually stimulate these gustatory cells. And then we have cranial nerves that are used or that are then stimulated to actually signal what our taste is. So the facial nerve, which is cranial nerve 7, the glossopharyngeal, cranial nerve 9, and the vagus nerve, cranial nerve 10, are all involved in the process of taste or the interpretation of taste. Moving on to olfaction, which is the scientific word for smell. So odor travels in molecules. These molecules bind to our cilia. Remember, these are projections or like those hair-like projections at the end of olfactory receptor cells. So we have receptor cells specific to smell. So once that molecule binds to our cilia, an impulse is then going to be generated along nerve fibers in our nasal cavity, and then this travels through that ethmoid bone, which is right kind of in the center of our nose, if you remember back from the skeletal chapter. So these fibers synapse with other neurons in the olfactory bulbs. So the olfactory bulb up here, that's where the actual nerve junction or synapse occurs. So these structures are right below the frontal lobe. If you remember back from a few slides ago, that olfactory center is kind of right on the bottom of the frontal lobe there. That's what this is talking about. And so once that synapse happens, the signal will then go to the primary olfactory cortex in the brain, that kind of little highlighted green area, if I remember correctly, right below the frontal lobe. Okay, now moving on to hearing. This is a little more in depth. The ear is a super important area, especially once we get all the way to the inner ear. So we talk about the ear in 3 different parts. We have our outer ear, our middle ear, and our inner ear. So we'll go through all parts of that. So the auricle is this outer lobe of your ear. And then you have this is your auditory canal. And so the auricle, the shape of it, and the auditory canal, the shape of it, the idea is that it's going to be able to funnel sound into this canal, which can then travel into the middle ear and inner ear. So we'll talk about sound conduction through the whole process. Okay, so our middle ear actually has 3 bones in it. These are the smallest bones in our body. And so they help transmit our sound basically through vibration. So we have the malleus, the incus, and the stapes. So malleus, incus, stapes, which then connects to our oval window. This you do not need to memorize, but the eustachian tube helps equalize pressure on the tympanic membrane. So that's part of the tympanic membrane vibrates in response to our sound waves. And so the tympanic membrane is what really separates our outer and middle ear. And as I said, when we have our sound wave, it vibrates, and that's what then transmits the initial vibration to the malleus, which goes through the rest of the inner ear bones. So the inner ear itself plays a role in hearing, but it also plays a role in balance and equilibrium. So our semicircular canals have a role in balance. And then our cochlear nerve are all hearing. The Vestibular nerve is your balance one. So we'll go into these structures just a little bit more in depth. Okay. So the organ of Corti, that's your hearing organ. So anytime we talk about the organ of Corti, you're thinking about hearing. So this is really a zoom in on the organ of Corti. So we have the basilar membrane. That's just the floor of that organ. Our cochlear duct is right at the start of that organ, and then the perilymph is what is filling the organ itself. The tectorial membrane has thousands and thousands of hairs that project from it. This movement is partly how the organ of Corti interprets sound. And the tectorial membrane is kind of like a gel. It's very flexible and moves along with those hairs. So which inner ear structure contains the structures for hearing? A, the vestibule, B, the semicircular canals, C, the cochlea, or D, the auricle. So think about that one. C, the cochlea. So let's pop back up, actually. I thought I had one more slide, but let me pop back up to this one. Okay, so as I said, the semicircular canals, you should think of equilibrium and balance. The cochlea has all of our structures for hearing itself. The oval window is where our malleus, incus, and stapes end up transmitting that sound. And the round window is the very end of the line. And I think I have a little bit more on the steps of hearing, so we'll talk about that in just a second. And then our vestibule is where our bony and membranous labyrinths are, and those are also related to balance. Okay, as I said, I had these steps slide that we'll talk about next. Okay, so the auricle helps guide sound into the auditory canal. The next stop is that tympanic membrane. Remember, that's our kind of border between the outer ear and the middle ear. The malleus is resting directly against that tympanic membrane. So when the tympanic membrane vibrates, this then triggers the malleus to vibrate, which triggers the incus to vibrate, which triggers the stapes to vibrate, and then this triggers the oval window to vibrate. So that oval window vibration causes the perilymph, which is kind of the filling or the fluid that is in this area, to shake or vibrate on either side of our cochlear duct. So this shaking is going to travel through the cochlear duct to stimulate the organ of Corti. Remember, that's our hearing organ. The organ of Corti is what interprets, and the movement of the hairs of that organ are what cause impulses to go across that cochlear nerve for our auditory, the auditory cortex to interpret sound. So the organ of Corti is our final stop prior to traveling to the brain. So eventually we hit the round window, and that's when our ripples dissipate. So the oval window is where our stapes connects to. The round window is the end of the line. Okay, so we talked just a second ago about how our semicircular canals in the vestibule are very important for balance. And now we'll go a little bit more into the detail aspect of that. So in terms of the semicircular canals, they have fluid within them. And so as this fluid moves, we are able to sense that, or our brain is able to sense that. And that's partly how it kind of keeps track of where our head is in space and how we are moving. So at the end of each of our canals is what's known as the ampulla. And so there's a lot of hair cells in here. And so as our fluid moves and moves those hair cells, that's how it's able to detect kind of our movement and keep us on track. So the ampulla is at the end of each semicircular canal. The vestibule can be further broken down into the saccule and the utricle. Okay, we just talked about this. This is our antler. This is at the end of our semicircular canals. We're not going to go into to a ton of the details here. I really just need you to know that the semicircular canals end in the ampulla, and the semicircular canals, the ampulla, and the vestibule, which includes the saccule and utricle, are involved in balance and equilibrium. Okay, so dynamic equilibrium and static equilibrium, there's two different aspects of this. So the speed and direction of head movements is primarily our dynamic equilibrium. This is done primarily by the semicircular canals. And then the static equilibrium is typically the utricle and saccule in the vestibule rather than our semicircular canals. So don't get caught up too much with dynamic and static. I don't think you need to understand the in-depth aspects between those two. You just need to know the aspects that make up our balance and equilibrium system. So the utricle and saccule reside in the vestibule. So the cupula is part of the ampulla. The cochlea has the organ of Corti, and that's our hearing organ. The ampulla is at the end of the semicircular canals. Okay, the second half or the last third of this PowerPoint is all about vision. So this is just a highlight of the accessory structures. And these are the definition of our accessory structures. So you should be familiar with all of these structures. I'm not going to read you each definition. It's relatively straightforward here. Okay, our lacrimal apparatus. So this is basically our tear ducts, but it's a little bit more in-depth than just a simple duct. So we have a lacrimal gland itself. That's what secretes our tears, which goes right onto the surface of the conjunctiva. So these can help clean the eye surface. They obviously keep it moistened, deliver oxygen, and it also has lysosyme, which helps prevent infection. It takes care of bacteria. The lacrimal apparatus also consists of the lacrimal punctum, which is a super tiny pore that's at the end of the lacrimal canal itself. And so that is kind of how our tears actually drain. We also have the nasolacrimal duct, and so that also helps drain our tears into our nasal cavity. So if you ever notice kind of the people who are what they call ugly criers, where they get all sad and sob and their nose gets all runny, that's because of the lacrimal punctum and the nasolacrimal duct, where it's draining into our nose. Okay, so the eye movement is controlled by 6 different muscles, which are innervated by 3 different cranial nerves. So the trochlear nerve, the abducens nerve, and the oculomotor nerve. So nerve 3, 4, and 6. So you do not need to know the numbers or the names. You should know that there's 6 total muscles, 3 total cranial nerves. So it helps the eye move right and left, up and down, but also rotate and move on the diagonal as well. Okay, the tissue layers of the eye. So our outermost layer is the fibrous layer. Our sclera and our cornea are both parts of this. So the sclera is a very dense connective tissue. It's the outermost layer total. It's the white of the eye, and that's where all our blood vessels run through. The cornea is a transparent, what they consider an extension of the sclera. It's very anterior. It covers the iris, which is the colored portion, and it has a role in admitting light that has no blood vessels. So all the blood vessels are in the sclera itself. The vascular middle layer that has our choroid, our ciliary body, and our iris. So the choroid is also a highly vascular layer, which is the primary kind of supplier of oxygen and nutrients. The ciliary body is an extension of that choroid, which ends up forming what they consider a collar around the lens. It secretes the aqueous humor, which we'll discuss in a couple of slides when we talk about the cavities of the eye. The iris is the colored aspect of the eye. And then we have our neural inner layer. So the retina is a super thin layer, but that's where a majority of our light sensitive cells are. And then if you follow out the back of the eye from the retina, you have our actual optic nerve, which is cranial nerve 2, that connects to the brain. So the retina has our photoreceptors. So these are broken into rods and cones. So our rods typically are the black and white, less detailed receptors, whereas our cones are our color and fine detail receptors. So if you look here, you have your fovea centralis. Our cones are concentrated there, so that's where our most precise and sharpest vision is. Our rods are kind of spread out everywhere else because they're more of a general receptor. And as I said, the nerves end up leaving the retina and converge to become the optic nerve. Where that convergence and exit occurs is known as the optic disc. So this is a bit of a description on what we just talked about there. Okay, the outermost layer of the eye is go ahead and look back if you need to, sclera. So remember, ciliary and choroid are that middle aspect, that vascular layer, and retina is even further in the neural layer. Okay, so we're looking superficial to deep here, not necessarily superior to inferior. So we're superficial. This is the surface, or looking straight ahead. This is deep. So the eye is broken into our anterior and posterior cavities. Within the anterior cavity, you have an anterior and posterior chamber as well. So our anterior cavity is between the lens and the cornea. So between this layer. The posterior cavity is posterior to the lens. As I just said, there's an anterior and posterior chamber to that anterior cavity, but the whole anterior cavity is filled with a clear, watery fluid that's known as the aqueous humor. So our lens itself, that's our dividing point. That's a transparent disc of tissue, and it lies behind our pupil. That's our, like I said, that's our border or dividing point between our anterior and posterior cavities. We'll talk a little bit further, but our lens can change shape depending on if we need close or far vision. So the posterior cavity is filled with vitreous humor. This is less watery. It's more of a jelly-like substance, but it gives our eyeball a little bit more structure so that it doesn't collapse. So the ciliary body itself secretes our aqueous humor. This fluid flows from the posterior chamber through the pupil into the anterior chamber. And then you guys really do not need to know this term, but as our aqueous humor recycles, it goes through the canal of Schlemm. You guys just need to know anterior cavity equals aqueous humor, posterior cavity equals vitreous humor. Aqueous, watery, vitreous, jelly-like, structural. Okay, the process of vision. This seems very simple. So light focuses on the retina, so kind of our lens and the other anterior structures direct the light onto the retina where our sharpest vision is, and it actually produces an upside down image. So our rods and our cones interpret and convert this image into actual nerve impulses, which are then transmitted to the brain for actual interpretation and for us to decide what we see. So the next few terms, you just need to know the general concept of. You don't need to know the ins and outs super detailed. So refraction itself refers to the bending of light rays. So vision all comes down to how our rods and cones interpret light. So our eye is structured, and the different eye structures are constructed in a way to manipulate and allow us to utilize these different light rays to interpret information. So our cornea and lens bend these light rays onto our retina and that fovea centralis so that we can interpret or get the best picture. So convergence is our next. So refraction, really the only thing you need to know, think refraction equals bending of the light rays. Convergence is the actual movement of our eyeballs. So when we're looking at something far away, it's fine for our eyes to stay parallel to get on the visual axis. When we get something closer, the light waves actually diverge more. So the eyes have to move inward to be able to get back on that visual axis. So convergence is the eye movement inward to attain our visual access, and this happens when we are having something that is nearsighted, or we're looking at something close to us. Okay, pupils can constrict and dilate. That's how we control the amount of light that's getting through. So the whole idea is that we constrict and dilate to reduce our blurriness or to focus in. And so this happens with the use of an intrinsic eye muscle. So there's actually a muscle that causes pupil constriction and dilation. So to constrict, the pupil or the pupillary constrictor muscle is stimulated by the parasympathetic nervous system. Our pupil then constricts and less light comes through. When stimulated by the sympathetic nervous system, our pupil dilates, and so more light comes in. So you see they have dilator and constrictor muscles. So constriction is parasympathetic, dilation is sympathetic. Okay, accommodation is the next concept that we're going to talk about, and this is related to the lens itself. So the relaxation or the contraction of the ciliary muscle controls the shape of the lens. But what you guys really need to know is that accommodation is the concept of the lens changing shape based on how far or close an object is. So basically, our lens changes shape so that we can focus on an object based on the distance. Okay, so just to dive into rods and cones just a little bit more, I know we kind of talked about this back when we were talking about the retina and fovea centralis. So rods kind of sit at the periphery of the retina. There are dim light receptors. Night vision relies on rods. They're not good at distinguishing colors. Cones are going to be in the center of our retina. Bright light is the trigger for our cones to kick in. Our sharper vision relies on cones. Color vision relies on cones. And actual integration of colors is related to cones. So people that are colorblind have a deficiency in how their cones integrate color. So this kind of talks about how our vision impulses actually cross sides, but this is a little unnecessary for you guys to worry about right now. Okay, the bending of light rays is called, remember we just talked about these concepts, A, convergence, B, accommodation, C, constriction, D, refraction. And the answer is D, refraction. That's the bending of light. Convergence is the actual movement of the eyes to get to that visual axis. Accommodation is the changing of our lens shape based on the distance of an object. And constriction is to restrict light entering the eye. And then opposite of that would be dilation, which would allow more light in. All right, that's the end of chapter 12. If you have any questions about this, please reach out, but don't overthink this stuff. This is kind of a pure memorization chapter.